This video is devoted to one of the most important applications of KaosKey's novel non-stationary biomedical signal processing technology, namely to the analysis of the human brain's electrical activity, the electroencephalogram or EEG. The recording of human EEG by means of electrodes on the scalp is a non-invasive brain imaging technique that provides a window into the functioning of the brain with excellent temporal resolution. The EEG time series displayed in the upper panel gives a visual impression of the EEG as a strongly non-stationary signal. The problem is that non-stationary signal analysis does not enjoy a well-developed mathematical toolkit. The non-stationary character of EEG data is essentially ignored by current EEG processing methods. KaosKey has developed a new mathematical approach and advanced tools of non-stationary signal analysis, which provide means to decompose EEG signals into functional elements with high temporal resolution. In this context, the signal processing routines may be regarded as a mathematical microscope. To demonstrate performance of the mathematical microscope, the animation is stopped. Now, the motionless blue line in the upper panel of the screen is a 2-second segment of the EEG. The mathematical microscope decomposes the EEG into functional components, i.e., elementary building blocks from which the signal is composed. Such building blocks, termed a quasi-Gaussian kernel, QGK, is displayed in the plot inserted to the left side of the middle panel. As a component model, quasi-Gaussian kernel is defined by a minimum number of degrees of freedom, just the three parameters: onset time, peak amplitude, and sigma, the shape parameter. The identification of quasi-Gaussian kernels is performed by a sequential procedure displayed in the upper panel on the screen. Each step produces quasi-Gaussian kernels illustrated by the red line. The parameters of identified components are shown under the plot. These parameters define the individual quasi-Gaussian kernel, displayed by the red line in the left side of the middle panel. Conceptually, quasi-Gaussian kernel is a model of the corresponding EEG component. To give a visual impression of remarkable modeling accuracy, the red line in the bottom panel of the screen shows synthesis of the EEG model using the running results of the EEG decomposition. These decomposition and modeling techniques offer powerful tools for real-time recognition of specific activity patterns in the time course of an ongoing EEG. Here is an example of the identification of specific EEG waveforms, the morphology of which is consistent with characteristic features of the prominent event-related potential P300. This potential is widely accepted as an objective marker of fundamental cognitive processes. Conventional methods of the P300 estimation is the ensemble averaging. In the averages of single trials, the P300 usually appears as a large, positive, monolithic wave. However, single trial analysis reveals a rich repertoire of subcomponents, the most important of which are the P3A and P3B potentials. Accordingly, a characteristic feature of a single trial P300 is a complex component composition reflected by multi-peak profiles of this large positive waveform. The results of the search of such elements in the time course of ongoing EEGs are illustrated in the upper panel of the screen. The blue line shows the ongoing EEG. The red lines show the composites of quasi-Gaussian kernels, which produce large positive double peak or triple peak waveforms. Such morphology is similar to the characteristic morphology of the P300 cognitive potential. The technique of EEG decomposition using KaosKey's mathematical microscope provides powerful tools for recognition and identification of specific activity patterns in the EEG signals. Such comprehensive real-time analysis of non-stationary EEGs and identification of various cognitive components may provide groundbreaking insights into the information processing systems of the brain and promote the design of real-time brain-computer interfaces.